Optogenetics, a technique which combines genetics with light stimulation, is starting to be used in cell and developmental biology to control basic biological processes. For example, we can specifically guide the movement of a cell in a certain direction. We can regulate cell signaling pathways by controlling the activity of membrane receptors or downstream effector proteins. We can also induce and prevent actomyosin contractility of a cell or tissue. We can control life-death decisions, such as triggering apoptosis upon light exposure. And finally, we can enforce a specific cell fate by the light-dependent control of cell differentiation. The biggest advantage of optogenetics, compared with classical approaches for controlling biological processes, is the spatial temporal precision it can achieve. Since we use light to trigger the system, we can, for example, specifically activate individual cells in a tissue. In certain experimental setups, we can even achieve subcellular precision, such as a specific induction of cell protrusions at defined sites. Just as important as the spatial precision is the fact that optogenetics enables us to activate a system at precise time points to investigate dynamic biological systems. By changing the frequency, amplitude, and duration of the activation pattern, it is also possible to directly modulate the kinetics of dynamic biological systems. On the molecular level, there are currently three major groups of optogenetic systems. The first known system was the channel rhodopsin-like photosensitive ion channel. This channel opens in response to light and facilitates flux of ions to change the membrane voltage potential of a neuronal cell. Photosensitive ion channels are widely used in neurobiology, and approaches based on this system are referred to as neuronal optogenetics. The second system involves proteins that undergo light-induced dimerization. Light exposure induces a conformational change in a photosensitive protein domain. Then, a specific interactor exclusively binds the photosensitive protein in its excited state. Using this light-dependent heterodimerization, a fused target protein can be recruited to the plasma membrane. The third class of optogenetic systems is based on photocaging. In the dark, the photosensitive protein of interest has an inactive conformation, usually because of allosteric autoinhibition. Upon light exposure, intramolecular unfolding caused by minor conformational changes activates the target protein or exposes dedicated signaling motifs. Applications which make use of light-induced dimerization and photocaging systems in cell and developmental biology are referred to as non-neuronal optogenetics. This e-learning course focuses exclusively on these more recently evolved systems that can be applied in essentially all cell types and model organisms. Optogenetics makes use of a defined set of molecular approaches. The most commonly used approach is the recruitment of a protein of interest tagged with a photosensitive domain to a specific anchor protein upon light activation. Using the same principle, a protein of interest can also be sequestered away from its site of action. Removing a protein from its natural localization into a different cellular compartment essentially removes this protein's function. Some photosensitive proteins tend to form big homomeric clusters as a result of light activation. If the target protein is catalytically active when present in high density, then clustering results in the formation of active signaling hubs inside the cell. Optogenetics can also be used to control gene expression. Transcription of a gene of interest can be regulated by making use of these kind of dimerization strategies. For example, the light-induced complementation of a split transcription factor or recruitment of a transcriptional activator can trigger target gene expression. Using a slightly different approach, it is also possible to directly control the enzymatic activity or binding affinity of a target protein. A conformational change in photosensitive domain, bioengineered in the protein of interest, can expose a hidden signaling motif or relieve the protein from allosteric autoinhibition.